Hi everyone, welcome to another session of Tarvos TNPSC coaching classes. Today here we are with a strategy video on seed science and technology. This video is very helpful to it will be very helpful to each and every one of you because we will be seeing the questions or the areas from which question comes mainly for the seed technology part. From our analysis, we could see that more than 10% of the questions come from unit 6 of seed science and technology. Okay. In this, we will be seeing, as usual, we initially start with the question paper analysis. In that, the unit 6 consequently every year there were more than 20 questions. Only in 2023 you can see that this is just 19. Okay, in compared to all other areas of C technology, the exam blueprint shows that more than 20 questions have been there from this section till last year. Okay, of which most importantly you should see that the statement type questions and direct questions are being asked. Okay, that in that the 28% of statement type questions will be confusing. Okay, because as C technology has many legal legislations and those kind of areas included in that. So it is very easy to make tough statement type questions. So everyone should be very much aware of those traps that may come and those very minute things that you need to focus while answering questions from seed science and technology. Clear? Okay, let's see the question split up. The usual pattern that comes is 42% of questions are direct questions. Okay, I hope those questions will be somewhat more than 75% of those questions will be simple also. Basically like the color of the breeder seed tag or the number of visits for field inspection. Such kind of direct questions or germination percentage or other formulas. So those the kind of direct questions can come which most of the students will answer, but the rest of the 28 will be very good quality statement type questions and nearly 18% of match the following questions. Usually that isolation distances, those questions are very frequently asked. Isolation distance of each and every crop. Okay, or the amount of seed that you should be submitting for each and every test and everything can be asked. Okay, such as the following questions are very common, and assertion reason and definition type questions are comparatively less. Assertion reasons type of questions are less asked nowadays in the exams. Only in out of 100 you may get 2 or 3 questions of assertion reason, that is less, but more of statement type questions are coming in today's exams nowadays for the exams. Okay, yeah. Okay. And coming to the topic wise in-depth analysis, you can see that this should be the priority of your study when you touch unit 6 of seed science and technology. And first area of most importantly and most commonly asked is the seed classes. This is a very easy area also, everyone knows, because you need to study the tag color of every category of seeds, from nucleus seeds to all other category seeds. This is comparatively easy, and don't take it lightly because this question is being asked continuously. Recurringly these questions are coming because that area is very important for the seed certification and the procedural part of seed certification. Okay, so that is very important, that's why it is frequently asked. Then comes seed testing. You should be very clear about the ISTA standards and the weight for each and every seed for testing. That's what I told you previously. Okay, for it can be very easily asked for match the following questions. Okay, it can be given as statement type questions. Any types of questions can be framed from that area. Okay, we will be going into the depth of each and every area, so I will be glancing over it once. Okay, then comes the vigor and viability. You may have done your practicals, everything in your lab, that like the Tetrazolium test, the color changing to red and other Vigour test. Okay, viability and Vigour tests are very important. Then as I told before, isolation distances, very important. For example, cotton has an isolation distance of 50 meter, while sunflower has around 400 meter. So it is not that easy to remember, but you should somehow refer Agarwal's book. You may be knowing that. Okay, Agarwal's book is very good for standard reference book for seed science and technology. So you should make a table of the uh, isolation distance of major crops. Okay, and study that well. Clear. And then the seed certification stages and the inspections. Okay, these are the areas from areas from where statement type questions can be asked. Okay, because these are procedures. From you won't be asked about the continuous procedural. You don't have any descriptive type of questions coming and all. So from you change the order of this stages. Okay, you change the crop. For example, there are 3 seed inspection in one crop. Other there may be 2. Change the crop name so they can easily confuse you. Similarly, such statement type questions are very common. Okay, then comes the Seed Act 1966 and the legal framework. This area, I would say that for the upcoming exams and all, this area will gain more importance. Why? Because there is a new draft published for Seed Act amendment. Okay, the old 1966 Seed Act is going to be changed. So the government has published a draft to take suggestions from all the stakeholders, from universities and everyone. So that process is in progress. So that is an area of focus nowadays for the past 6 months or so. That area, Seed Act area and their amendments, the rules and regulations in that, the different sections, mainly the different sections in that are gaining importance. So questions can also come from those areas. Okay, you can expect more importance to Seed Act and its proposed new amendments. So whenever you study Seed Act, no, don't just study the old 1966 Seed Act. Other than that, just understand what are all the new changes which are proposed to come. Okay, there are different changes in the portals. There are difference in the violation penalties. Okay, there are many areas that are being changed. We don't have the time to discuss everything of that act now, but whenever you go for the in-depth classes, study that part also in extra. Okay, because those things will not be available in your book. Okay, because these are very recent information. So that is very important, right? Then comes the moisture content and the, because moisture content and the weight etc are very important, because this play a major role in the keeping quality of seeds, or for maintaining the viability of seeds, or for controlling the pest and disease incidence of on the stored seeds. So moisture content is a very important parameter. So how which all are the methods you follow to study the moisture content? How much should be the moisture content of different seeds for different terms of storage? Okay, those things are very important. Then comes seed priming and seed sowing treatments, pre-sowing treatments, because seeds can be having different kind of fungal and other infections. It can be seed borne or from other surroundings. Things can happen. So you have many methods to prime these seeds, or these are activities done to improve the germination of the seeds. There are many terms related to seed priming and different techniques in seed priming that we will be just glancing in the later slides. Okay. Okay. Followed by that, there comes the seed processing machines. Each processing machine relies on different parameters of the seeds. So that is very important. Which parameter, which machine is focusing on, and what is the principle behind that seed processing in that machine? Which purpose, which kind of seeds can be used in those particular machines? Those kind of informations you need to be very clear. So the machine, the purpose, the principle behind that, which seed can be used. These things should be given importance. Clear? Okay. Then going to the next part, from which usually questions are less, but questions came from, that is purity analysis and the seed composition standards. Then hybrid seed production. In the previous strategy video of crop improvement also, we told about the importance of hybrid seed production. There are many systems, the male sterility systems, okay, which are helping in hybrid seed production. GMS, CGMS, we have 2-line system, we have 3-line system. Okay, these systems are very important. Okay, and the varieties released using those technologies are also very important. Okay, we already discussed that in the previous strategy video on crop improvement, right? Okay, then comes seed dormancy, which is comparatively, it is a simple area compared to other legal areas and other isolation distance and all. Seed dormancy is a very easy area. But there are some terms you should be very clear of, the stratification, scarification, the difference between those terms. Things are very simple here, very easy to study and understand. Then comes the seed biology or the seed botany. Okay, this is more of a botany side than that of agriculture side, but still there are some terms that you should be familiar with because they are very much associated with reproduction. Okay, sexual reproduction of flowering plants. So for example, you terms like nucellus, anatropous ovule, perisperm, okay, triple fusion, double fertilization. How the seed biology changes, okay, and the sexual reproduction related terms, okay, which leads to seed development, okay, and the different types of ovule arrangements. Okay, different types of ovule arrangements also you should be very clear with. Okay, then comes the next one, that is the weed seeds and objectionable contaminations, because these are parameters that are to be regulated, okay, very well monitored and regulated so as to ensure the purity of the planting material. Okay, so there should not be any objectionable weed. Some countries are even banned. India is banned to import things from some countries. Okay, and then you have some contaminants similar, which are physically similar to your seeds. Okay, for example, Argemone mexicana, okay, the poppy seeds and in your mustard and all are physically similar. You can't very easily detect them as the contamination. Okay, so that is very important. Weed seeds and objectionable contaminations, which are all the contaminations and which are all the seeds or plants which should be avoided during hybrid seed production or seed production. Okay, there are off types that is to be removed. That is the usual steps. Okay, these are basic things, but I am telling for some crops, some of the objectionable weeds and other similar seed producing plants are also there. They should be avoided in your field area. You should focus on those areas also. But these areas are not usually tough because very few points are there. You can very easily remember. Then comes the Tamil Nadu specific seed production data. Okay. In South India, Karnataka, Telangana and Tamil Nadu are the large producers of seed material. So data changes every year very drastically. So it is very important that you should have an updation of how much seed production, what is the percentage of in the Indian scenario, how much percentage of this particular crop seed is produced from Tamil Nadu, or how much metric tons of vegetables are produced. Vegetable is produced from Tamil Nadu in the previous year. Okay, usually you should focus mainly on 25-26 data. Whichever year your exam is coming, you should focus on then just the previous year and the current year overlapping data. You should have a very clear idea. Okay, hope you got that priority list. Okay, clear with everyone. Okay. Now we will be jumping into a minor few minor details of each and every priority area we just went through. Clear? Okay. First of all, the seed classes. That was one of the very important and the most regularly asked question area, as we told before. Okay. That means there are different categories of seeds. This is well arranged in the same order. You have nucleus seeds that is controlled by the breeder, which is highly pure, followed by the breeder seed. Okay, that is having a golden yellow tag, while nucleus seed doesn't have any tag because it doesn't come to the public, right? Because the breeder itself is holding that small quantity of the purest seed, while breeder seed that comes out to the research station level and the scientists supervise its production. Breeder seed has a golden yellow tag. Then comes the foundation seed. As the color indicates here, it has a white tag. Okay, it is under official supervision of certifying agencies and the staffs associated with that. Then comes the certified seed, which is having an azure blue color tag. Okay, this is sold to farmers and this is multiplied and sold to farmers in the large level. So you should have a clear idea about the order of seeds, the different tags of seeds, which seeds are maintained by whom, and which is used by farmers. Clear? This area is very important, and understand few other key points that the seed village concept, they are only uh, advised or they are only allowed to produce certified seeds. Okay, foundation seeds and breeder seeds, they cannot produce foundation seeds and breeder seeds. Okay, seed village cannot produce that because seed village is a participatory program to produce large amount of certified seeds so that it can be distributed to most of the farmers or all farmers in the villages. Clear? Yeah. So we should be very clear about the color of the tags. This is the most usual question. Okay, hope that is clear. Then comes the sample size based questions. I told you, for each and every test, for example, germination test, purity test, moisture content test, the amount of seed that the customer or the person, farmer should give for this testing differs. Okay, for each test there is designated amount of seeds to be given. Clear? So this shows the, for germination test you can see that there are 40 or 100 seeds needed, while for purity analysis in paddy you need about 400 grams in case of paddy, and for moisture content you need 100 grams. So these kind of things are very important. So there are, for each and every crop there are minimum sample size for each and every test, and similarly there are minimum sample size for each and every crop. Got it? For example, here I have given the example of paddy, bajra and everything. Paddy you have 400 grams for purity testing, for and jute you can for grow out testing, for the Vigour grow out testing you need 500 grams of seeds, and for moisture analysis you need 100 grams of seeds in most of the varieties. Okay, so whenever a question comes, okay, see the data or 400 or that digit along with the unit. They may give 400 grams for germination test. No, it is number for germination test. While 400 seeds for purity analysis is wrong. It is 400 grams for purity analysis. Why we are telling or focusing is people become overconfident that this is a very simple question and we jump into a false answer. You don't see that unit kind of things. So give important to when you read a question, read it very thoroughly, slowly and completely before jumping into your answer, right? These minor mistakes can come to you, so keep that in mind. Okay, then comes the seed vigor tests, different seed vigor test. Okay, there is direct seed vigor test, direct number based test, Okay, and there are other tests like seed vigor index based tests. Both seed vigor test and seed vigor index are can be called as test for seed vigor. Okay, most commonly followed that seed viability or viability testing is by using tetrazolium chloride. Okay, this is the fastest method. Okay, you need not have any controlled environment. All your live cells will stain red color due to active dehydrogenase activity. That means whenever any cell is alive, it will respire. Okay, when it respires, dehydrogenase activity will occur, and that due to that activity the color changes. Clear? Okay, this is an indirect test for vigor, while it is a direct test for viability. Keep that in mind. Even though we are discussing on vigor, this tetrazolium test is mainly for seed viability, whether it is live or dead. While it is an only indirect measure for seed vigor. Because why I am stressing is such questions come as statements. Okay, such questions comes regularly as statements and people make mistakes. Okay, hope it is clear regarding the tetrazolium test. The detailed test and every chemical compounds involved in that and all you will discuss in our detailed class sessions. Those are also very important areas. Okay, right. Then coming to the next step, that is accelerated aging test, where the seed is given a stress. Okay, stress and actual germination percentage is calculated. This method, accelerated aging test, can be called as a real direct estimation of uh, seed vigor. Clear? Because you are giving a stress to the seed, still the seed is germinating, means that means the seed has that vigor to grow. Hope this is clear to everyone. These the points associated with that also is very important. Then comes the seed vigor index. There are two types of seed vigor indices. Okay, seed vigor index is of two kinds. One is length based approach and second one is direct seed weight based approach. Okay, so there you have questions, you usually has questions where the SVI1 will be having the details of SVI2 and SVI2 will be having the details of SVI1. Similar confusing statements comes. For example, SVI1 is based on direct weight method, direct weighing method, or based on seed weight. If that is a statement, that is wrong. Okay, so you should be very clear. There are two indices, that is SV1, which is a length based approach, SV2, which is a weight based approach. Okay, I have given the equation also for that. Here you need the shoot length after the germination, the shoot length and root length after particular number of days, while here you will be taking the weight of the seedling after drying it. Okay. Okay, these are the two types of seed vigor indices. Okay. Indices are also indicators of how much vigor the seedlings has, okay, or the seed has, while accelerated aging is a direct testing, tetrazolium chloride test is a indirect method of testing seed vigor. Hope these points are clear to everyone. This is, I am repeating, this is not a detailed lecture on seed vigor testing. We have many other more points to discuss in the detailed sessions. This is just to glance through the most importantly asked areas. Okay, you just use this session for prioritizing areas. If you have less time, mark these areas, mark these points to these areas for revision and complete its revision first. Okay, that is the purpose of this video, right? Okay, the next one is the isolation distance. As I told you, each crop has each different isolation distance. Okay, so more amount of outcrossing, more cross pollinated the crop is, there will be more of isolation distance. Clear? So how isolation distance is very important when the seed inspector comes to the seed production plot, because that is the first thing. The presence of off types of or other similar varieties and the isolation distance is the most important parameters seed inspectors will come and look at. Okay, and for that is very important. Here also I am saying that you refer Agarwal's textbook. Okay, or else you see to the TNPSC our Tarvos coaching, because we will be referring that book only. You make a table of different crops and revise it thoroughly. Stick it on your wall, because it is very easily you will forget these numerical numbers. So stick it on your table study board or your table itself, and then revise it continuously. You should see that multiple times, then only you will you will be remembering that. Clear? Okay. Then coming to the field inspection counts, this is also very important. For different crops, you have different number of field counts, field visits, samples. Okay, everything based on your area. Also, sampling changes. More area, more number of samples per acre. How much samples? Okay, those kind of things also are very important. Clear? Then about the hybrid seed production and yeah, grow test. One more important point: grow test is a prerequisite before certification. For getting certification, you need grow test result, positive result. Clear? So for each and every crop, what all you will focus? Isolation distance. Okay. Then this one, the number of field counts, which are all the how much quantity of your seed materials need to be taken, and the inspection times, which are all the stages of the crop where inspection from the seed inspector happens. Okay, these points you should, here it is just 3 crops. There are many crops, as you know, so you should be thorough with the other crop data also. Clear? Okay. Then coming to other, I won't say less important, but still important areas, that is seed priming and dormancy breaking treatments. Because seed priming, I would say once more, seed priming is any method that is done to improve the seed germination. Okay, so you do many techniques to improve the seed germination, and two terms that I need to clarify with you is the difference between osmo priming and hydro priming. Okay, both of the terms usually people confuse. While osmo means, here osmo priming, osmo means it's just solution, not water. Hydro means water. If you are clear in this, then you won't make a mistake. Clear? Here in osmo priming, you use different solvents for controlling the rate of germination. You get a uniform, healthy amount, healthy normal germination rate after osmo priming. While hydro priming is more of a simple thing. You just soak the seeds. You take the seeds and just like in your home, you just soak that in water, and then you go for sowing that. Okay, that way people do very commonly. That is hydropriming. You are just allowing the seed to imbibe the sufficient amount of water they need for germination. While osmopriming is more of technical term. It is a controlled germination, controlled favoring of germination by using agents like polyethylene glycol, PEG, and other sorbitol and other different chemical solvents. Clear? Yeah. This is the difference between the hydropriming and osmopriming. And the PEG, as you know, it will not penetrate the cell wall. Okay, it is tough to penetrate cell wall. Usually, PEG is given to seedlings for stress studies, drought stress, and all. You give PEG treatment. If the seedling survives that, we call that seedlings are somewhat tolerant to some stress like drought. Okay, right. Then other things, dormancy breaking strategies, scarification and stratification are 2 methods. There are many other methods that I am repeatedly telling. This is not a study class. This is just to understand the main areas, or to go through the main key points usually people make mistakes of. Okay, difference between stratification and scarification. Here, scarification is a very simple thing. You just rub the seeds on a rough surface and make a small scar on the seed coat. Then through that seed coat, the water and other things will be imbibed and the seed germinates. This is usually done where in some crops where the seed coat is really hard, while stratification is different. It is giving chilling stress, moist chilling stress to the seed, okay, for some weeks, and then the seeds will start germinating. This is done when the seeds are having a dormancy. Okay, one is done to break, scarification is done to break the physical dormancy due to hard seed coat, while stratification is done to overcome physiological dormancy by giving this stress like cold stress treatment. Clear? Dormancy is also of different types. We are not going to the classification now, but still you have physical dormancy, you have chemical dormancy, you have physiological dormancy, you have many kinds of category of dormancy itself. For each and every dormancy breaking, you have different techniques. So that detailed classes you should be very attentive, and you should be studying from those detailed classes also. Clear? Okay. Here in the base of it, you have very commonly in hard seeds are very common in black gram. Okay, for that you go for scarification. Even if at your wild oats, all you have all the different types of dormancy together in this one crop, so it is very tough to germinate. Okay, then acid delinding of cotton is a very common technique used in cotton. Then you go for hot air treatment. As I told you before, there are different methods to overcome different dormancy. Okay, that is very important. Okay, you need to refer to the detailed lecture for studying that. Okay, right. Then coming to the seed processing machines, I told you, you need to hear what all things you need to study. First of all, the machine name. Second, what parameter or related to seed it measures. Third, which mechanism and which parameter and which mechanism that machine follows. Okay. Fourth, which all crops this machine can be put to use. Okay, you can't use same machine for this one mustard and for rice. Okay, which crops are suited for using in which machine. Okay, these four things are very important. Okay, and for example, I have seen these are the 4 points you need to study regarding this. Okay, one example I have seen shown here. Here you see disc separator. It is based on length of the seed, while gravity separator is based on the density of the seed. Okay, cyclonic separator is also based on length. The Bornier divider is mainly based on the gravitational force. Principal or parameter, machine name, which seed it can be applied to, okay, and the mechanism by which it works. These 4 parameters you should be very thorough with. Okay, hope this is very clear to everyone. Then coming to the next one, that is the purity and germination standards. Another very important area, similar to your isolation distance. Okay, you have purity standards for different class of seeds. Questions usually come from certified seeds. For example, you can see minimum purity standard for paddy is 98, while for groundnut it is 96. Okay, minimum germination standard, it is 75% for pearl millet, while 65% for cotton. So I am saying that you should make a table, isolation distance, purity standards, minimum germination standards, okay, of all these crops, and see that list regularly, because only by seeing it every day you will be able to remember these numbers. Okay, You should be very clear, because if statement type question comes, a simple question can come that like this can come, the minimum purity standard of black gram is 75 percentage. No, it is regarding black gram, it is 75 percentage, but that too it is germination percentage. Clear? Such confusing statement type questions can be very easily asked. Okay, such kind of questions can be expected, right? Then coming to the breeder seed status and everything, yeah, okay, if you know about the different class of seeds and how they are produced, then this area can be also be covered. Okay, so initially, as I told you, you should have a clear hierarchical order for the seed production. Each seeds has different kind of tags, that also you should focus. And here these numerical values also you should be very thorough with. Here things are more to be mugged up, I would say, and not don't take in a bad sense, but compared to other subjects, you in seed science, you need to by heart a lot of things. You have by heart a lot of procedures and datas. Okay, that's why people get confused also. So they know to how to confuse you. So you should be thorough with these numerical datas. Clear? Yes. Okay. Then coming to the Tamil Nadu based seed production data. Here you can see for few examples are given. For Bajra seed production, it is nearly 22 acres should yield nearly 9,000 heads. Okay, for Italian millet, okay, seed yield is 10 to 16 quintals per hectare. Similarly, for major crops also like rice, cotton, and for such major crops also, you should be having the state average yield or say state average seed production rate. Usually, the cultivation practice and seed production, excuse me, yeah, the cultivation side and the seed production data has a few differences. Okay, usually very easily you will get the production data, while seed production data is not that easy to get. So whenever you study, don't confuse with the production data and seed production data. Okay, production data is the amount of that particular produce. Okay, for example, paddy production is this much, that means this much tons of paddy was produced in Tamil Nadu state. That doesn't mean that whole thing is seed. That is for consumption purpose also. Clear? So how much amount of seed is produced for each crop, that is important for. But in the general side, they can also ask the same question in your same exam. But from seed science prospect, the seed production rate or the whole seed production of your state, Tamil Nadu, need to be given more focus. Clear? As I told you, you should, these things you won't get it from any standard books. Okay, you need to have a data of clear updated data of the previous year till 23, 24 or 25, 26 at least. You should have a clear list of these numerical datas, statistical production datas. Okay, these things you should take your own effort, because there are lot of crops. Okay, there will be a lot of data, just like it is like GK only. You will be getting those information and you should study that. Okay, these things will be included in our classes also, but you should update yourself these kind of things. Clear? This production data and everything you should be updating by yourself, right? Then coming to Seed Act. Yeah, coming to Seed Act, I told you this is an important area currently, because the amendment of Seed Act is being proposed. Okay, so in our classes, detailed classes, we have discussed the different sections of Seed Act. Few of them are given here. Okay, for example, section 5 gives the details of on varietal notification, while section 4.2 deals with the seed testing lab standards and how to establish that. Okay, then there is sections that regulate or deals with the penalties, sections regarding the extension, regarding the extension of time and duration of or extending the certification period. then sections regarding the certification agencies establishment, and then sections containing the power of the seed inspector. Okay, you should know the section number and what that section deals with. Okay, and in addition, as an updation, you should know the newly proposed seed act and the differences it has from the traditional seed act. Hope that is clear, right? Yeah. Then coming to the moisture content part, this similar to your the seed viability index, moisture content can be also measured in two ways, that is a fresh weight basis and dry weight basis. Most students in your practicals you may have done this seed moisture content analysis. That is very common, you know, that is fresh weight based and dry weight based. You should be clear about the formula. Okay, things are very simple, but you should be very clear with the formulas, right? Okay. Then seed botany, I told you before, in seed botany you should be very thorough with the different terms. For example, perisperm, it is given here, the diploid maternal tissue from the nucleus. What is nucleus? Nucleus is the nourishing cells for the developing embryo. What is funicle? Funicles is the it is the stalk of the whole ovule setup. Okay, and the integuments which covers the embryo sac, right? There are antipodals. You need to have a clear idea regarding the structure of the ovary. Secondly, then the different arrangements of ovule, different kind of inflorescence, different kinds of seeds, and different kinds of fruits. Okay, there are different types of fruits, achenes, berries. Okay, there are many types. For example, in the inflorescence you can see caryopsis, you can see this one, panicles. Okay, you can see arrows. Okay, there are different types or terms related to botany. Okay, you just study fruit, seed, seed development, how that embryo is developed and the structure regarding that. Okay, these are the major importance you should focus on. Clear? Hope this is clear to everyone. Okay. Then comes hybrid seed production and the seed multiplication ratios. Okay. Hybrid seed production we already discussed in our excuse me. Yeah, hybrid seed production we already discussed in our previous classes and all. Here also we will discuss it once more, because hybrid seed production involves different strategies like male sterility. Okay. A line, B line, R line, and these lines are all involved in the hybrid seed production using male sterility systems. Okay, that we already discussed, and you should have a clear idea regarding the different types of male sterility systems, which of them are used in hybrid seed production, which all lines are involved in each type of systems. For example, CGMS is a 3-line system. Okay, GMS, while this TGMS is a 2-line system. Similarly, the synonymous names are also very important. Clear? Yeah. Then comes hybrid seed production related terms. Okay, for maintaining the quality of hybrid seeds, there are many techniques followed, like detasseling. Okay, or removal of off types. Okay, there are many methods followed, and there are many breeding related terms also. Because mainly you see in crop like maize, there are many methods for quality seed production. You should be focusing on those kind of methods, breeding methods for developing hybrid seeds in specific crops. Okay. then comes the seed multiplication ratio. Okay, groundnut is a very common question. 1:8 or 1:10 is the seed multiplication ratio of groundnut. While in case of paddy, it goes very high because it is 1:100. Because one paddy seed can produce more than 100 seeds. Okay, for example, tomato, if you see one tomato seed give rise to the plant, and each plant produces thousands of seeds. While in case of groundnut, you see, one seed will produce only 8 to 10 seeds. So people also do research accordingly. You know that, okay? Because whenever in breeding experience, if with one cross is success, you will get in the next generation you will get a large population if the seed multiplication ratio is very high. So you can see more of research is done in crops which are having high seed multiplication ratio. Okay. There can be a doubt why we are studying all these things. This is a purpose. As a seed technologist or a breeder, you should be having a clear idea of the seed multiplication ratios. Okay, here you study the seed multiplication rate of major crops, that is enough. Okay, and those crops which are having highest and which are having the lowest. Okay, hope this is clear to everyone. Then comes the ISTA standards and the wheat seed contaminations. Okay, here this is just a very superficial information. Okay, first of all, we will see ISTA because that is an international seed testing association. Okay, or the standard setting as or setting body. Okay, which is mainly located the headquarters in Switzerland. Okay, and this they published journals named Seed Science and Technology. Okay, first seed testing station is in Germany. Okay, you understand that. Then ISTA seed dormancy breaking of tomato is done with potassium nitrate. Few key points that were asked in previous years are just given here. You also need to study the ISTA standards, which are all the standards that ISTA has set for ensuring the seed quality. Okay. Then comes the most one of the important part, easy but important part thing is the weed seeds and the contaminations. Because whenever you produce seeds, it will be having some or the other contaminations. So, seed inspection and the seed quality control organizations are maintained or kept very strict monitoring for the contaminant identification. Clear? Okay. For example, contaminants can be of different ways. It can be internal, it can be physical contaminants. Okay, it can be internal because some fungal infections or something can come. So you should be having, you refer your theory class of Tarvos and see the categorizations of contaminants. Weed seeds is one of the contaminant. Okay, while other than weed seeds, you will be having the dust and other debris type of contaminants. Okay, you will be having in your plot. You will be having rogues. Okay, unnecessary crop plants, everything. So all these are contaminants in seed production, of which weeds and pathogens are very important. Weeds are objectionable weeds. There are weeds which are considered objectionable weeds, weeds which are not allowed in few particular crop hybrid seed crop seed production plots. Okay, so that those things you should be very clear with. Okay, right. Then yeah, this is the comparisons, usually asked comparison uh, type of questions. You should be very clear with, as we already discussed all these things, but this is just to give you an outlook how question comes in the exam. The difference between foundation seed and certificate seeds, what are all the differences, who maintains that, what is the color of the tag, those things are very important. What is the purity percentage? Okay, then we seed foundation seed, likewise all the type of class of seeds. Okay, then the SU1 seed vigor index 1 and 2, we already discussed the difference. One is based on the seedling length, while other is based on the dry weight of the seedling. Okay, then fresh weight and dry weight based moisture analysis, the germination and purity analysis. And in addition, we already also discussed that the viability and test and the vigor test difference, viability and vigor, which are all the direct and indirect tests, such things. Then sampling, okay, moisture level and the number of quantity of seeds and the moisture percentage, you should not be confused. Osmo priming and hydro priming difference we have already discussed. Okay, right. Then the difference between different machineries. okay, in improving the seed quality, okay, the machinery purpose seed and the type of seed it use, what is the principle there the machine use, everything you should be very clear. Then the isolation distance, okay, of each and every crop. Then the objectionable weeds and other contaminants in each and every crop. This is also a very important area. Okay, once you cover the areas that we discussed today, you cover this unit completely, because these are the only areas from which questions came from. So you should be a smart worker than a hard worker. You focus on these areas, finish it off, and then go to the next unit, because revision is the most important thing. Studying is not the matter, revision matters. More you revise, more score. More in depth you study, you will not be helpful, because you will forget things. Okay, we are all human beings, we will forget. More you just glance through the topics, okay, like C technology where concept is less, points are more things to by heart than to understand. Okay, C tech is that kind of a area. Unlike breeding, in breeding you need to know many concepts. Okay, I won't recommend this kind of a study method for that. But still, in C technology, you can glance through the things, then again glance through it, give multiple revisions, then only you will be remembering that numerical data and the examples, everything. Okay, right. And finally, before concluding, we will just once more see to the important points. Vigour test, color of the tags of different crops, different types of seeds, priming, the different kinds of priming, machineries and their uses, hybrid system. Already we discussed in plant breeding crop improvement unit also, that is also very important. ISTA standards, sampling size, isolation distance, and the different objectionable weeds and other contaminants. Okay, then moisture percentage and other formulas related simple direct questions. Okay, dry weight basis, wet weight basis based simple formulas. Then CDAT 1966 and its amendments and details on each and every sections in a very concise manner. Okay, then field counts and field inspections. And also you should have an idea on the different stages of seed certification. Okay, that I forgot to inform you, that is also an important part. What are the different steps to get certification for a seed or a variety? Okay, right. These are the main things that you should follow. These are the main areas you should not skip before you go to your exam hall, because this unit, this is just again the same questions asked as in a question level. After at the end of the class, you should be at least able to answer correctly regarding these questions at the least. Okay, so main thing is unit 6 of seed technology is that important. That's why we arranged a specific session for this. Okay, nearly 10% of your questions in your question paper is from this area. Okay, never skip this. If you have less time, give this section the highest priority in C technology. Okay, hope everyone is clear with the important areas. Okay, just see this video in a very fast manner. Okay, don't waste much time. Okay, understand the key areas, keep a screenshot of the area. Okay, and then cover each area, tick mark each area, finish it off and move to the next unit. Okay, be a fast learner and a smart learner than a hard worker. Okay, that's the strategy to crack the exam. Okay, all the best for everyone. Study well and crack your exam and come out with flying colors. And Tarvos wish you all the best. Okay, from my side personally also, I wish you all the best. Thank you for attending the class and make a good strategy and execute it well. Okay, thank you everyone.